Will Norton is a PhD in developmental biology from University College London. After his PhD graduation, he studied his postdoc in EMBL Heidelberg, where he worked on zebrafish fin development. After this, he pursued his postdoctoral research in the Helmholtz Center Munich and CNRS in France on the topic of zebrafish behavioral neurobiology. And he is now a lecturer and associate professor in animal biology at the University of Leicester in UK since 2012. And his research focuses on zebrafish neurogenetic behavior and welfare. So we are looking forward to your presentation, Will.
Thank you very much. Thank you for inviting me to speak this afternoon. I'm sorry I've not joined most of the rest of the meeting. I've been teaching all day. I have to teach later as well, so I can only pop in to give my talk today. I'm just uploading the slides via the PowerPoint Live, so you have to bear with me a few moments, please, while it loads into Teams. It's nearly there.
No worries. We're happy that you managed to join in between of your lectures, and also most of the talk are actually recorded right now, so you can have a look at them later on.
Fantastic, thank you. Can you now see my slides okay?
Yes, all good.
Wonderful. So the title of my talk today is Quantifying the Potential of Skin Swabbing as Refinement for DNA Sampling of Laboratory Fish. Many animals which are used in scientific research need to be identified at some point in their life, and a typical way to do this is to genotype the animal by collecting a small DNA sampling and using that to amplify genes. The typical way this is achieved in zebrafish and other small laboratory fish such as sticklebacks is to use a fin clip, in which the fish is first anaesthetized in a tank until it's no longer touch responsive. A small part of the tail fin can then be removed using a sharp scalpel, and that piece of fin tissue can be processed to get some DNA out of it. And fin clipping is a really good source of DNA. You get high quality DNA at quite a high concentration. However, there are some indications that fin clipping can be harmful for fish. It can cause them pain, as well as altering their physiology, locomotion, or behavior. And that can be a problem if you need to use these animals in subsequent experiments, such as measuring behavior. It also increases the risk of infection from the wounds created by taking the fin clip. And that suggests there's a need to refine the way in which we can collect DNA samples for fish. This is actually very important because if you look at my slide, unfortunately I changed the picture this morning, it's a little bit blurry. But these are statistics from the UK Home Office in 2021, showing you the percentage of animals used in research. And the red circle shows you that 15% of experiments are now carried out on fish species. The largest number of animals are rodents such as rats and mice at 64%, and fish are the second largest category at 15%. 85% of zebrafish labs worldwide use fin clipping to identify their animals, with over 3,000 facilities each year genotyping hundreds to thousands of fish a year. So that means there's potentially millions of procedures being carried out, and that could be painful for these animals. Together with other colleagues in the University of Leicester, so Ken Wintilly and Carl Breaker, we tried to adapt a technique called skin swabbing to collect DNA from fish species. The skin swabbing procedure was developed by other researchers for use on larger fish. We adapted it for these very small animals, zebrafish and sticklebacks. The skin swabbing procedure is relatively easy to perform. You can catch a fish in a normal hand neck. I'll start my film again so you can watch this again as I'm talking. You catch the fish in a hand net and restrain it gently on top of a wet sponge without using anaesthetic. You can use the side of the net to hold the fish still, and then use a rayon tip swab to collect a mucus sample from the side of the fish. So here we can see Kenwyn has caught a wild type zebrafish in a net. It's a little bit hard to see, but hopefully you can see she strokes the side of the fish with the rayon tip swab. The fish's mucus will contain sloughed off cells from the skin, and you can extract DNA from these skin cells. So the swab has been now placed into a DNA extraction buffer, and we use simple laboratory techniques to get DNA out of the sample. Using this technique in a first paper in 2017, we directly compared fin clipping and skin swabbing. What we could see is that we had very high quality DNA if you look at the OD values for both fin clipping and skin swabbing, but in both zebrafish and sticklebacks we had a much lower concentration of DNA for swabbing compared to fin clipping, as shown here. However, we were able to recover enough DNA from our skin swab samples to PCR and identify animals. As an example here, I can show you a cross-contamination experiment. We wondered whether fish held at high density would bump into each other and swap mucus samples between them, which would mean you would see cross-contamination of the DNA recovered. In order to investigate this, we took two groups of zebrafish, a wild-type line and a line which expressed GFP under the control of a promoter, and we crowded them together in a small tank overnight at a higher density than we'd normally use for stocking. We were then able to identify both GFP and a second gene called MITFA from the GFP line. That's shown on the right-hand side of my slide here. I turn my pointer on, you can see that's up here using the highlighter. Whereas in the wild-type fish, we could amplify MITFA alone, but we didn't amplify GFP, suggesting that no cross-contamination had occurred. Having established that we could use skin swabbing to collect DNA samples, we wanted to know whether it really was a refined technique to collect DNA from fish compared to fin clipping. So was it less stressful for the animals, for example? In order to do this, we undertook a study funded by the NC3Rs here in the United Kingdom, and we compared three groups of fish. We had control fish which were allowed to swim around undisturbed in their home tank. We then compared a standard skin swabbing procedure in which the fish were captured in a net, held in the air briefly as they were transferred to the sponge, and then swabbed before being returned to their home tank. The third group of animals were captured, anaesthetized, and underwent a standard fin clip procedure before being allowed to recover. We looked at different readouts of welfare in both zebrafish and sticklebacks. But for the sake of time today, I'll simply show you the zebrafish data. In fact, they were quite similar across species. The first readout that we looked at was release of the primary stress hormone cortisol. And cortisol is released when the stress axis is activated in response to an external stressor. In humans, for example, cortisol will increase our heart rate and make us sweat if we're very nervous. We were able to measure cortisol by the excretion of this hormone into the water. The fish underwent either a fin clip or skin swabbing procedure. We then placed them in a glass beaker with system water for 30 minutes, and we could extract the hormone from the water using a SEPAC column. By doing this, we were able to show that there was a heightened release of cortisol following a fin clipping procedure compared to the control fish or the fish which had undergone skin swabbing. And we believe this is mostly likely due to the pain caused by removal of the fin tissue. You can see this on the graph on my slide here. On the y-axis we have the concentration of cortisol released, and on the x-axis we have the three different groups. You can see that the skin swab fish in the centre look fairly similar to the control fish, whereas the fin-clipped animals tended to secrete more cortisol. It's worth noting here that we saw quite a wide range of results between the different fish species. Some of the fin-clipped animals appeared to not react to this procedure and didn't release much cortisol, whereas others released quite a lot of the hormone. The next readouts that we looked at were changes in behaviour. We looked at a number of different behaviours, but today I'll show you locomotion, or total distance swum in 5 minutes, and time spent at the bottom of a tank, which is a readout of a thick metaxis, a preference to spend time at the bottom or the side of the tank. And we know from numerous other studies by other researchers that anxious animals tend to swim at the bottom of the tank, whereas less anxious fish swim up in the water column. On my film here, you can clearly see two different groups of zebrafish. Some of them are much more anxious, they're remaining at the bottom of the tank, whereas the less anxious animals are swimming up in the water. We looked at the changes in behaviour 30 minutes after either procedure, skin swabbing or fin clipping, 1 day later on day 2 as the fish began to recover, and then 1 week later on day 7. to see how long it took the animals to recover from these two different procedures. In general, we saw a very wide range in behavioural responses across the fish which we measured and made it quite difficult to see patterns or changes in behaviour in the different groups of animals. And you can perhaps see this by the size of the box of whisker plots I've shown on my graphs here. However, if we first focus on day 1, we saw a reduction in locomotion, the total distance swum by the fin-clip fish compared to the skin-swabbed and the control animals. On day 2 in the middle here, we see a reduction in locomotion in the swabbed animals compared to fin-clip and control. And on day 7, there was a reduction in both the skin-swabbed and the fin-clip fish. So it's complicated data, and it shows that the happiest fish were those which were simply left undisturbed in the tank, with different changes over time. We saw a similar pattern in changes of behaviour when we looked at time spent at the bottom of the tank in a novel tank, which was our anxiety test. On day 1, the fin-clipped animals spent more time at the bottom of the tank compared to swabbed animals or controls. On day 2, it was the skin swab fish which spent more time at the bottom of the tank compared to the 2 other groups. And by day 7, we saw no differences in behaviour across the 3 different groups. The final readout of stress we looked at was expression of stress marker genes in the brain. We looked at a range of 6 different genes, and once again, for the sake of time today, I will focus on 2 of these for you. Brain-derived neurotrophic factor, which is also called BDNF, and corticotropin-releasing hormone A. We use BDNF as a general readout of stress, and has been shown to respond to stress levels in both zebrafish and other animals. Corticotropin-releasing hormone A is one of the components of the stress axis, which is activated when cortisol is released. In general, we reasoned that a higher level of gene expression would indicate a more stressed zebrafish, whereas a lower level of gene expression is perhaps harder to interpret. It could represent a less stressed fish, or you could argue that any change from homeostasis from the control level acts as a reduced readout of welfare. So let's look at our results over time once again, focusing on day 1, 30 minutes after the procedure, on day 2 and day 7. On day 1, if we look at the expression of BDNF, we can see heightened expression of BDNF in the fin-clipped animals compared to the control and swab group, and this is significance once again. On day 2, we can see that it's the skin-swabbed animals which show heightened expression of BDNF compared to control or fin-clip. And on day 7, once again, both groups of fish appeared to recover. There was no significant differences on either type of animal, although I do note the greatest spread of data in the skin-swabbed animals on my graph on the top right-hand side here. We also looked at corticotropin-releasing hormone A. Once again, fin-clipping quickly upregulated expression of this gene, and you can see a significant difference on day 1 in the fin-clip group here compared to control on swab. However, on days 2 and 7, there were no differences between the groups, regardless of which procedure they'd been through. So no difference in CRHA in fin-clipped and swabbed fish compared to control on day 2 or day 7. So in summary, it appeared that using either technique wasn't particularly good for the fish, but in general we saw a larger number of changes when fin-clipping the animals. One of the ways in which the fin-clip procedure can be improved is by applying an analgesia. An analgesia which is typically used is lidocaine. There have been a number of studies that have shown that pre-treatment with lidocaine can reduce the harm caused by fin-clipping. We wanted to know whether we could further improve our skin swabbing technique by giving the fish lidocaine 45 minutes before we swabbed them. In this case, we used 4 groups of animals: a standard fin-clip without lidocaine, or a fin-clip with lidocaine pre-treatment. We also looked at skin swabbing both without lidocaine and with pre-treatment with lidocaine. And in this case, we looked at changes in behavior using an automated system called the Fish Behavioral Index that was developed by Professor Lynn Steddon at the University of Gothenburg. The Fish Behavioral Index, the FBI, is an automated system that gives a readout of changes in locomotion and space usage in the tank. And as such, it gives you a composite score to try and work out whether you've seen changes in fish health and welfare. We also looked at cortisol release once again because we saw such striking results without lidocaine use. In general, we found that pre-treatment with lidocaine does not improve the swabbing protocol, and that's shown on my slide here. And this data was collected by Lynn for us as part of a collaboration. In the fish which underwent a fin clip on the left-hand side without lidocaine, we saw a significant release of cortisol. If we pre-treated similar animals with lidocaine and then fin clipped, we saw a reduction of that release, and we didn't see any difference between swabbing with or without lidocaine treatment. Finally, we used the Fish Behavioural Index as a readout of behaviour, and the FBI generates a score between 1 and 0 to tell you how healthy your fish is. A score between 0.84 and 1 means that the fish are healthy. A score between 0.67 and 0.83 is an okay animal. Unhealthy fish will have a score between 0.33 and 0.66, and a severely abnormal animal will display a score between 0 and 0.32. Once again, we compared both fin clipping and lidocaine with and without pre-treatment and fin clipping and skin swabbing with and without pre-treatment with lidocaine, and the results here were once again very striking. In the absence of lidocaine pre-treatment, the fin clip fish showed a severe reduction in the FBI score. It started at 1 hour, became very apparent at 2 hours, and the fish was still very abnormal 6 hours later. Compared to this, if you treat with lidocaine, the fin clipping looked very similar to a skin swabbing procedure with or without lidocaine pre-treatment, and all of the animals had a score at or around 0.9 or 1, suggesting that they're healthy. So in summary, so far we compared skin swabbing to fin clipping, and we saw some differences between the two techniques. The fin clipping causes a statistically significant increase in cortisol release, suggesting that the stress axis has been activated, whereas the swabbing doesn't, even without using anesthetic pre-treatment. We saw general increases in qPCR markers. We saw a greater number of qPCR markers change following a fin clip compared to the skin swabbing procedure, although, as I've already said, the happiest fish were those control animals allowed to swim undisturbed in their tank. Finally, in general, in our hands, we found that behavioural readouts were a rather poor way to look at welfare because we saw a very large variation between different animals in the different groups, perhaps because of the way in which we designed our behaviour experiments. It was difficult to see differences between the groups, but in general, we saw greater differences after fin clipping on day 1 and then skin swabbing on day 2. So far we've shown you that swabbing appears to be a refined technique to collect DNA compared to fin clipping, even in the absence of anesthesia or analgesia. The swabbing technique is easy once you've learned to perform it, and it's quick to perform, and you also don't need to use sharp scalpels to collect the tissue, so there's a reduced risk of cutting yourself when you perform this. We're currently being awarded some more NC3R's funding to try and continue this work, and we wish to look at using different types of swabs to see whether we can reduce the changes we see in fish post-swabbing. We're also going to look at the damage to the fish's skin caused by swabbing by removal of the mucus layer and how quickly that could recover over time. And finally, we're interested in investigating whether we can adapt this to use on other species that also have a mucus layer, such as frogs and toads. So thank you very much for listening to me today. This work was all carried out at the University of Leicester, working with my friends Ken Wintilly, Hector Carreño and Carl Breaker. We've had long-standing collaborations with Professor Ian Barber at the University of Aberystwyth and Professor Lynn Swadden in Gothenburg, and support from all the fish team here in Leicester. And we're very grateful for funding from the National Centre for the Replacement, Refinement and Reduction of Animals in Research here in the UK. Thank you very much.
Thank you so much, Will, for your very insightful talk. Are there any questions from the audience? Feel free to write it down in the chat, in the Q&A, or raise directly your hands, please.
Thank you very much for this very interesting talk. We already had a small glimpse of it in Lynn Sneden's talk.
That's perfect, yes, great.
And I'm very happy that you provided a applied refinement method, already something that is already there to implement. There was actually one question from the audience during Lynn Snedden's talk. I will ask it. I don't know if it has already been answered, but it says, I was wondering how much the PCR genotyping is efficient after swab procedure to collect DNA.
We have success in around 90% of the swabs which we've collected. I would say it's fairly similar to the fin clip technique in that we didn't always manage to PCR from fin clipping. I think there's a slight learning curve. I think you have to get used to swabbing the fish. For example, being the PI, I'm sorry to say I've rarely done this. I would ask Kenwyn or Hector to do this. They're much better at the technique than I am. But I think once you're confident at doing this and at extracting the samples, it should work as well as the fin clip. The only real difference I can see, as I've already noted, is the smaller concentration of DNA that you can recover. Kenwyn and I have been talking to automatic genotyping companies. So you already have companies where you can take fin clips and post them off and they process the samples for you. We've been trying to develop the swabbing so that they can use swab samples as well. It looks reasonably promising so far. It does work, but the companies haven't yet got it to that same level of accuracy that they see in the fin clips. So we're still working on that, and we can't roll out commercial or the automatic genotyping using this yet, but that's one of our goals.
Okay, but it can be used. It is useful, and
yes, we don't fin clip anymore. We've given up entirely.
Okay, and from what I hear when I talk to colleagues, there might be a little hesitation to go to skin swabbing because they fear it might be more difficult to teach to the students how to do it. But as you say, there is some learning curve, but do you think it's much more challenging or not at all?
Oh, no, not at all. I think they're probably equally difficult. The hardest thing I always found with fin clipping is to get the anesthesia right. And anesthetic hasn't been terribly well studied in small fish. It's been co-opted from other animals. So getting the dosage right isn't necessarily that easy. In fact, you don't have to use analgesia or anesthesia. The thing you have to learn is more a manual how to hold the fish. And I think the first times of catching it and holding it gently in the net, it does feel a little bit difficult. But with practice, you can get better at that. So within the UK here, Kenwyn and I go on sort of road trips and we go to other universities and we simply help them do it in the first cases so that they get over that first hump. I'm like everybody else. If something works in my lab, I don't like to change technique because I simply want to get on and do the experiments. And we hope that by going and actually helping people hands-on in other universities here in the UK, that will help them get more familiar with it and be confident in adopting it in their own laboratories, for example.
Okay, great. So there's another question from the audience. Starting at what age or size can you use the swabbing technique on a fish? Are they sometimes too small to give enough DNA material?
So we're very fortunate that the NC3Rs has recently given us some more money to pursue questions like this to further refine and establish the technique. In the original paper by Carl, who was our fish tech, and he really led this study, he was amazing. We said the smallest type of fish you can use is 20 millimeters. In fact, we don't really know whether that's the case or not. We simply think that the smallest fish Carl could find swimming around was 20 millimeters. So one of the things we're doing at the moment is trying to look at smaller and smaller fish to work out the smallest size limit we can use, not to hurt the fish and to get enough DNA to genotype. So at the moment, we published in 2017 20 millimeter body length. We're trying to work and refine that. Yeah.
Okay, thank you very much. So you are a lot there are a lot of thanks for a great talk.
Thank you.
Including some more questions. I think we have time.
There's 2 more questions in the chat. Yes.
Yeah, exactly.
Is it okay if I answer them? I'd be delighted to.
Okay.
Yes.
Yeah, we still have 5 minutes. All good.
Okay, so first question comes from Chloe. She asked about there are commercial products you can buy which you place in the water, and these are particularly used after fish transporting larger fish to help fish recover from wounding. One example would be something called stress coat. Within one of our papers on this, we compared whether post-treatment in these compounds you can dissolve in the water improves recovery from the skin swabbing technique. We looked at aloe vera alone, we looked at stress coat, and one other, the name escapes me. Unfortunately, it didn't work. It didn't improve the technique in zebrafish or stickleback. In fact, we found that zebrafish were somewhat less happy after we treated them with these commercial products. We think that's possibly because they've been designed for other larger fish species. They've not been optimized for the really small laboratory fish that we're using. But I wouldn't recommend, based upon our results, that we use these post-treatments in recovery stuff in water. Have I considered doing a video of the swab technique to place online? There's a question from John Scarpa. Yes, there's a NC3R's resource here. So if you go to the UK NC3R's page, they have a whole website which includes our official, if you like, protocol. It includes the video I've just shown you, which I made on my smartphone when doing this, but it's not an optimal video. So within the money we've just been awarded, we're going to make a professional video with someone who actually knows what they're doing. That's not me. So there is a video you can look at online, but it can be improved. And I'm hoping that the new properly made video will make it much easier to see what we're doing. Yes. Tess McCann asks, do you use specific commercial products for DNA extraction, or do you use homemade materials? In the first paper from Carl and myself, 2017, we compared different ways to extract DNA. In our hands, we found the cheapest way was to use the homemade buffers ourselves. Subsequent to that, other researchers have published they've been using some commercial kits, but we found it simply worked as well, if not better, simply using standard molecular biology techniques. There's a paper published in F1000 in which you have a version of our protocols, an official protocol, if you like, which explains exactly how we do it here. It doesn't mean it can't be improved, but it explains where we are with it at the moment.